At this time, I'd like to ask Dr. Robert Carr to come in and introduce our next speaker. Dr. Carr is a Southwestern University graduate and a physician. He's currently the chairman of the Board of Trustees of Southwestern University. He previously led the Research and Development Strategic Management Group at Pfizer Pharmaceuticals. He continues to work as a biological consultant and entrepreneur. Dr. Carr.
Thank you, Ben. Hello, everyone. Thanks for being here for this wonderful program. So Southwestern is all about lifelong learning, as you know, so I want to start by telling you about something I learned today. I learned that our next speaker's last name is pronounced Davis, which was not my first inclination from reading it, but I learned that from a reliable source. So Dr. Davis has spent more than 2 decades in senior positions in science publishing as an editor, author, and publisher. Now, lest you think that editors of scientific journals don't have a sense of humor, I want to share with you something that I learned on the internet that he has said about himself. He said, "My greatest contribution to biomedical research was hanging up my lab coat about 20 years ago and realizing that the only way I would see my name in the likes of Nature Medicine is if I snuck in the back door and joined the editorial staff." So I would say that insight has benefited the field of science publishing greatly. Prior to his outstanding career in science publishing, he received a master's degree in biochemistry from Oxford University and a Ph.D. in molecular genetics from the University of London. Now, science breakthroughs, like we've heard about earlier today, find their way to others through multiple routes. And Dr. Davis has made significant contributions in at least 2 of these ways that information gets to others. First, through leadership roles in science publishing with notable positions, including the founding editor of Nature Genetics, the first Nature spin-off journal, founding editor of BioIT World, editorial director of Cell Press, and in his current role as the Vice President Business Development at the American Chemical Society and the publisher of a well-known publication called Chemical and Engineering News. The other category in which he's contributed is as an author, and this is another way that scientific breakthroughs find their way to broader audiences through the kinds of books that he's written. And those include the first one called Breakthrough, The Race to Find the Cancer Gene, that was published in 1995. Cracking the Genome, Inside the Race to Unlock Human DNA, which was published in the year 2000. And then more recently, the $1,000 Genome, The Revolution in DNA Sequencing, and the New Era in Personalized Medicine, which was published in 2010. So please join me in welcoming Dr. Kevin Davis.
All right, thank you very much. Good afternoon, everybody. It is a huge pleasure for me to be here in Georgetown, Texas at the 36th Brown Symposium. We had 2 fantastic talks this morning on slow medicine and nanomedicine, and so I've got a very, very tough road to follow here, but I'm going to try and tell you some of the incredibly exciting things that are going on in genomic medicine and introduce you to the joys of learning about your human genome. In addition to telling you about the revolution that is going on in DNA sequencing that's going to bring us, and indeed arguably has brought us, to the brink of the $1,000 genome, I'm also going to hopefully serve as a warm-up act for the second speaker this afternoon, Anne West, with whom you will hear about the joys of really going on a genome journey into your personal genome. So this is what I want to talk about. This is obviously a whimsical cartoon about looking into a crystal ball, and the cartoonist would have you believe that your DNA can sort of reveal about as much about your future health and well-being as the crystal ball. Whimsical, yes, and I'm certainly not going to suggest that genetics is the answer to all of your ills. But I will point out a number of examples where advances in genomics are coming not just to you in terms of an inquisitive individual learning about your personal genome, but increasingly are having a major impact on the way we practice medicine. And in keeping with the theme that we heard this morning, and we will again tomorrow, hopefully helping, even though this is brand new technology, the technology is becoming increasingly affordable. That's one of the major themes of my book. And hopefully those savings will be passed on in the forms of cheaper healthcare as we end a phrase that I'll say probably a couple of times. We start to see an end to what people are calling diagnostic odysseys, scenarios where patients are in hospital receiving test after test, genetic panel after genetic panel, as doctors sort of take educated guesses as to what might be wrong with them. We now have the ability to just look at your entire DNA in one fell swoop in order to ferret out the potential mutations or glitches in your genetic code that might be predisposing you or giving rise to a particular disease in your loved one or family member. So you had a lovely introduction about my background. This is where I did my PhD in North London. This is just down the road from Paddington Station. This is St Mary's Hospital Medical School. It's famous for two things. Number one, as you see from the plaque on the screen, this is where Sir Alexander Fleming serendipitously discovered penicillin in the 1920s. And this is also, you're thinking, where have I heard about St Mary's in the news recently? This is where all the royal babies are born, the royal heirs to the throne. William and Harry and George, I think George was the last one, I think. And of course, this was greeted with all kinds of media fanfare and glowing, gushing newspaper headlines last summer or whenever George was born. My favorite was this one. I think Private Eye really got it just about right. So this is St Mary's Hospital. I went up to do my PhD here in 1983. We were trying to find disease genes. We were trying to find genes that cause horrific genetic diseases like muscular dystrophy, cystic fibrosis. Colleagues of ours were working on Huntington's disease and Alzheimer's disease. Incredibly important pieces of work. And we didn't have a clue how to go about it. We had no roadmap. The human genome was almost a complete mystery. This is one of my favorite photographs. This is what human genome looks like. You have 23 pairs or bundles of DNA, all beautifully color-coded, like you see here, just like M&Ms. Okay, we've used a little bit of computer wizardry to dress them up here. But this is the map. This is our universe. And as we were tried in the early 80s, mid-80s, to find where we might uncover the genes for cystic fibrosis, for example, your guess was as good as mine. We didn't know if it was chromosome number 1 or 17 or 12, or as it proved to be in 1985, number 7. We had no encyclopedia, we had no sequence. All we could do was, it's a bit like throwing darts at a dartboard and hoping you hit a bullseye. We would isolate tiny fragments of DNA that could serve as signposts, and we could track how these markers were inherited through families. And if we got really lucky, and one of those markers happened to sit fairly close to the disease gene that we were trying to ferret out, in this case the CF gene on chromosome 7, we would see that the pattern of inheritance of our marker sitting nearby the disease gene would follow almost 100% the segregation and the inheritance pattern of the disease gene. So this is how groups like this one and many others map the genes for Huntington's disease and cystic fibrosis. But it took millions of dollars, thousands of hours of manual labor, redundant effort as groups in Boston competed with Houston, competed with San Francisco, competed with London, and it was clear that by 1990 we had to have a better way. And that's how the Human Genome Project got started. That's how people like Jim Watson, the man who co-discovered the structure of DNA, were able to go to Congress and say, we've got it, guys, we need a roadmap. You've got to spend some money, $3 billion would be nice, to chart out over 15 years a roadmap of the human genome so we don't spend years and years of taxpayer money frivolously trying to map one gene. We've got to just build a map of all 20,000 or however many genes there are. The next big race of this type, the one that led to the first book in my extensive bibliography of 3, was the race for the breast cancer gene. And you heard about Angelina Jolie and her personal story as a carrier of the BRCA1 gene mutation. The gene was mapped, just like I showed you on that color-coded genome map, to chromosome 17 in 1990, and then a furious race emerged between the woman who did that work, Mary Claire King, who was at Berkeley at the time, and a biotech company called Myriad Genetics in Salt Lake City. And I think many of you know how that story ended. By a matter of weeks, Myriad found the gene just before Mary Claire King and her colleagues were able to isolate it. And of course, as was their prerogative, they were able to patent it. And the ramifications of that patent continue to roll on. There was a major decision from the Supreme Court handed down just at the end of last year in violating most gene patents, but Myriad still has other patents that it thinks it can defend, and so even though a number of other biotech companies have quickly come out to offer alternative tests for the BRCA1 gene that Myriad previously had a monopoly on, Myriad is now suing the heck out of them in the hope of preserving its legacy in the breast cancer genetic testing department, all the while sitting on a trove of probably thousands, if not tens of thousands of mutations that are their proprietary intellectual property. I show this because that book somehow found its way to a film director a couple of years ago who called me up and said, "We'd love to get your help in making a film. We're making a film about one of the first women in North America who was tested for the BRCA1 gene," and that film was made. And it's just in the last couple of months received a distribution deal and should be coming to a theatre somewhere near you sometime this year. And many of you will recognize the principals in this. This is Helen Hunt, of course, famous TV actress and film actress, playing the role of Mary Claire King, which is a sort of a side story to the main narrative of the film. And if you look at the movie poster there, yes, that's Jesse from Breaking Bad in a great role. So thrilled to be a part of that. So back to the Genome Project. 1990, the effort gets underway. It's a sort of a loose collaboration between a number of academic groups. And by the halfway point of that 15-year voyage to sequence the entire human genome, by the halfway point, we just about managed to cobble together about 2 to 3% of the sequence. It was going pretty slow. I won't get into the reasons. It's all in the book that you can buy later. And then this guy entered the picture. This is Craig Venter in a photo image taken in about 1998, 1999 at the, I was going to say the command center. It almost looks like the bridge of the Starship Enterprise, doesn't it? And I think that was this was a biotech company that he founded called Celera Genomics, and their aim was to mount a private takeover of the Human Genome Project. They felt they had the better machines, the better technology, and the better strategy to, for hundreds of millions of dollars rather than billions of dollars, just put all of our DNA in a blender, sequence the heck out of it, and then let a computer, the world's largest civilian supercomputer at the time, put the massive jigsaw puzzle together. And if they could do this while the NIH-funded and Department of Energy-funded groups were still cobbling together their stuff, these guys would have a premium on potentially priceless data that Big Pharma would pay millions of dollars a year to gain access to and beat out the competition. And that model worked for a short time, but as detailed in my book, Cracking the Genome, the NIH, and with the considerable help of the Wellcome Trust in the UK, a major philanthropy, major medical supporter in the UK, upped the funding, bought a ton of the same machines that Celera had, and began to sort of play catch-up. And that led, I'm skipping over a lot of colorful but somewhat transient details here, that led to this somewhat awkward announcement in the White House by President Clinton in June of 2000, declaring a tie between these two groups, the NIH and the Department of Energy International Consortium, represented by the tall guy, the smiling visage of Francis Collins, now the director of the NIH, of course, and the rather more gloom-struck figure of Craig Venter, who's probably because his share price was starting to come down, and he was thinking, how did we not get to celebrate this ourselves? Tony Blair is beaming in via satellite from 10 Downing Street because the Wellcome Trust and the scientists funded by the Wellcome Trust at the Sanger Institute, I'll get to the origins of that name in a minute, did about 1/3 of the total sequence. So that was June of 2000. That was just an announcement. Where were the papers? The papers came out about 8 or 9 months later, One published in Nature magazine, that was the NIH consortium effort, and Craig Venter and his colleagues published in the same week in Science magazine. These, of course, are the two most prestigious science magazines in the world, Nature published in the UK and Science published in Washington, D.C. The 5 individuals that you see painted on the cover of Science represent supposedly the anonymous, ethnic backgrounds of the 5 hitherto anonymous individuals who donated DNA for the Celera genome sequence. As you may know, about a year later, Craig Venter went on 60 Minutes and put his hand up and said, "Hey, it was me, I was the one, I put my sample at the front of the queue." Why not? Who wouldn't do that if you had been in my position? An interesting sort of corollary to that, though, is, well, so what about the Human Genome Project? Who does that belong to? Well, the donors for that, also anonymous, were recruited by this advertisement that appeared in a Buffalo newspaper in 1997. And you're scratching your head and you're thinking, why Buffalo? Well, it's because I had one Breaking Bad reference. Here's my second. The cook, the molecular biology whiz kid who could make better high-quality DNA libraries than anybody else, 99.9% pure, was based at Roswell Park in Buffalo. So that's why they recruited volunteers from that part of the world. Unfortunately, that means that your human genome could well be somebody like that. There are times when these two groups who were feuding for many years try to make up, and they receive awards and they shake hands politely, but if you scratch the surface, these old rivalries still very much come to the surface. Craig Venter here saying we did the first genome in history, the first complete version of the human genome, which was actually his a few years later. And you can read all about that in his latest book that just came out recently. And here's Tim Hubbard, one of the senior computer guys in the working with Francis Collins and colleagues, saying that we won. We won because we released all of the data into the public domain as we were generating it. That's what this was all about. We made it public, and Celera wanted to keep it somewhat closed because they were trying to run a business. So I'm not going to say much more about the Human Genome Project, other than this was the official announcement. April 2003, NIH puts out a press release saying, we've completed the Human Genome Project. Geez, isn't that great? There was no paper to accompany this. It was all a little bit contrived. It had a sort of a mission accomplished feel to it, I rather feel. And 2 weeks later, there was George on the aircraft carrier. Mission accomplished. I'm sure this is where he got the idea. Why April 2003? Because 50 years earlier, in April 1953, Crick and Watson, Francis Crick and Jim Watson, published the classic double helix, the iconic structure of DNA. Just a truly iconic piece of work. I'm not going to show that paper. Instead, I'm showing a letter. This is the most valuable letter in the world, in history. This went on auction last year and sold for $5.3 million, smashing the previous record. This is a letter that Francis Crick wrote to his son Michael when Michael was 12 years old in a British boarding school. And this is really Francis's first, as far as I know, first effort to sort of describe his building excitement that he and Jim Watson were about to make history with the publication of this structure. Our structure, Michael, is very beautiful, he wrote. DNA can be thought of roughly as a very long chain with flat bits sticking out. He used slightly more elaborate language in the Nature paper. And then he went on and he drew it. And so this is really, I think, the first ever drawing. This is all images from the Wellcome Trust library. First sort of hand-drawn picture of the double helix. And then he ended the letter, "Lots of love, Daddy." And God, thankfully, Michael didn't sort of, as you know, most 12-year-old kids today would go, "What the heck was this?" and throw it away, and kept it. And half of the proceeds of the letter went to the Salk Institute in San Diego, where Francis Crick spent many productive years at the end of his life. He passed away some time ago. The paper in Nature was published in 1953. It took nearly 2 months for the New York Times to get wind of that breakthrough. And that was the story in the New York Times. We heard about slow medicine this morning. This is sort of slow medicine. But we didn't have internet and things like that. What does the human genome look like? I present to you the human genome in printed form. A group at the University of Leicester, for reasons best known to themselves, decided we'd like to see and show people what a human genome absolutely looks like. So in very small font, single space, double-sided, this is your human genome, all 3 billion bases, all these 4 letters, A, C, T, and G, those are only the 4 building blocks of DNA. This is what it looks like, and they're color-coded by chromosomes, similar to the color codes that I showed you a few minutes ago. My favorite chromosome, in a way, I suppose, is the Y chromosome. This is, of course, the critical chromosome that differentiates men from women. And a couple of decades ago, a brilliant female geneticist named Jane Gitschier at UCSF had a stab at making up a map what the Y chromosome should really look like if you were able to map the genes. And if you just cast your eye over some of these traits, while I grab a sip of water, I think you will agree. Who needs genome projects when you have female intuition? That's what I say. The Human Genome Project, obviously, as I've just described, we were able to get a draft of the full sequence of our DNA. We got it in a little bit under the 15 years and a little bit under the $3 billion, but it was still, that's a lot of work, a lot of people, a lot of money. All of that work was done using a chemistry for sequencing DNA developed by this man, Frederick Sanger. He passed away at the end of last year, living into his 90s. One of only 4 people to win the Nobel Prize twice. He invented a method for sequencing the building blocks of proteins in the 1950s. And then for an encore, he decided, I think I'll turn my attention to nucleic acids and see if we can't do a method for that. And the method that he established, called dideoxy sequencing. That's just a kind of a variation of the DNA building block that he put into the chemical reactions to sort of spike it, and then you would track the different chains being built using some radioactive label. That method worked from when it was published in the 1970s all the way through the Genome Project into the early 2000s. And this method is still used in many clinical labs and forensics labs around the world. It's been superseded, as I'm about to tell you, but this is still in many contexts the gold standard, and it's just a remarkable accomplishment by this brilliant scientist working in Cambridge, England. That's also a little significant. So, okay, we're getting the Human Genome Project done, but we can't spend another $2 billion for a second genome and another $2 billion for a third genome. It was clear, even before that White House ceremony, that we needed some new methods. We needed to do for DNA sequencing what we've done for computing power, as you heard from Mauro this morning, turning the compute power that could send a man to the moon in a massive installation in Houston, Texas, or wherever, into the size of an iPhone. That's what we needed for DNA sequencing. And that's what a number of groups set out to do. And in 2002, I heard for the first time this expression, the $1,000 genome. And with millions and billions rolling in my head for the cost of sequencing a human genome, the fact that somebody would have the gall to say we could and should aim to develop a technology that can give us a genome for $1,000. I mean, this sounded like fantasy land. But this was Craig Venter pushing this idea. And regardless of your feelings for his role in the Human Genome Project, there's no doubt that he is one of the more successful and visionary scientists in the United States. And when he says something like that, a lot of people are going to take it fairly seriously because he's shown he has an ability to sort of see forward and see where technology is going. So in 2002, he has a non-profit institute. He brought his annual conference to Boston and held a symposium and invited half a dozen scientists onto the stage to talk about their technology solutions or the potential solutions to bring about in 5, 10, 15, 20 years, the $1,000 genome. Some of those people are still highly successful in the genomics world. Some have pretty much disappeared. One of the companies that presented was a small British company I never heard of before called Solexa. And these are the two hard-drinking, intellectual founders of Solexa. They're both professors of chemistry at Cambridge University, Cambridge again, Shankar Balasubramanian and David Kleneman. Here they are having a celebratory pint at the Panton Arms. I'm very fond of the story, just going back to DNA for a second, where just before that Nature paper was published, after Crick and Watson had built the famous model of DNA, there's a story that many of you have heard probably, where they ran into a pub. It was called the Eagle Pub in Cambridge, and this one lunchtime, and Francis blurted out, "We've discovered the secret of life!" And I would love to imagine that there was some punter at the end of the bar looking up from his beer and saying, "Oh, shut up, Francis, you idiot. God, you're always doing this," and went back to his drink. So that was the Eagle. This reunion, however, took place at the Pantanals because this is the chemist's pub. The Eagle apparently is the biologist's pub. This is the chemist's pub. This is where the real heavy drinking and scheming goes on. And Shankar told me that in the very late 1990s, he had an all-important group meeting to build the concept for his company, Solexa, here at the Pantanals. I said, "Well, why do you have it at the pub? I mean, I know you like a beer, we all do, but why would you do that there?" He said, "Because I swear," he said, "we have no office space, we have no seminar rooms, our department is so old." It's the chemistry department at Cambridge University, for God's sake. It's 500-600 years old or something like that. So they had to go to the pub. So they conceived a company based on a technology that these two guys were developing for sequencing single molecules of DNA. And when I say sequencing, in a way I mean the reverse. What they're really doing is they're photographing the growth of DNA molecules. And as each base, A, C, T, G, C, G, C, 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 gets added in, gets stitched into the molecule, you take a photograph and you can kind of then, you're watching the sequence be built. You're not sort of breaking it up, but you're watching it sort of in reverse. This is what that technology looked like in the early 2000s, and in a way still does today. These are small beads. They're really just sort of clusters of identical DNA molecules, and you hopefully can see 4 colors if the projection is working reasonably well. I think it's so. You can see the green, the red, blue, and yellow. And each of those, this is one snapshot in time as a series of nucleotides are washing over the DNA molecules here, and whatever the last base to be added to these different molecules are, you're able to see. And the way that this company worked, the way the technology worked, is they did it such that only one base could be added at a time. They played around with the chemical structure of the building blocks so that the base could be stitched in, but then it couldn't receive another base, not until you did another little bit of trickery. So you take a photograph, you then do that little bit you fix the end of the molecule so another building block can come in, and you keep photographing the fluorescent dyes that are tagged to each of those building blocks, and then you can build up the picture of the DNA sequence. And you're doing this on a microscopic, maybe not nanoscopic, but a microscopic scale, which is a complete breakthrough compared to the gel-based system that Fred Sanger and his colleagues were using. This is an email from 2005 that was kindly shared with me by Clive Brown, who was one of the senior scientists at the time at Solexa. It says, "We've done it." And they had sequenced, this was a breakthrough one weekend in February 2005, they had sequenced their very first genome. Okay, not a human genome, the tiniest possible viral genome you can imagine. The same genome, interestingly, perhaps deliberately, that Fred Sanger proved his method on some 25, 30 years before. And so he sent this email around saying, "We've looked at the data, we've done it." And I think most groups at this point, they would be popping champagne and they would be patting themselves on the back and saying, "Well, okay, where do we write the paper? Nature, Science, New England Journal of Medicine, what's the most prestigious journal you can think of?" And these guys did the precise opposite. They did nothing. They said, we don't care about academic glory and the papers and the press. We've got a job to do. We've got to get a box built and out and launched commercially. And so we're just going to stay razor-focused on doing that. And all the accolades and the journals and all that stuff, if we get around to that later on, okay, we'll do it, but not now. So this was early 2005. Meanwhile, back across this side of the Atlantic in Connecticut, they introduce you to this guy. This is Jonathan Rothberg. This guy lives large. This is a man who thinks nothing. He's been a successful biotech entrepreneur through the 90s, and he thinks nothing of excavating and importing 700 tons of Norwegian granite across the Atlantic to build a sort of a Stonehenge-type monument in his backyard. This is just south of Guildford on the Long Island Sound, for pity's sake. So if you can do that, you can build a sequencing machine. You're clearly, wired in a way that's, willing to think large or small as the case may be. And so Jonathan, about 5 years earlier, had spun off a company called 454. He never revealed what the 454 actually stood for. I make some guesses in the book. A method with some similarities to what Solexa was doing, but arguably further ahead, because in the summer of 2005, they published a big paper in Nature, just like Mara said, I'm only showing Nature papers. I'm pretty much only showing Nature papers too. He published, and quoted in an interview with me, he said, we won the race. I mean, I can't deny the evidence in front of me. I've got a paper out in Nature with a machine, and we're sequencing DNA. It's going really well. Where's the competition? There was no sign of it. There's the paper. The lead author, the first author, had worked on the Hubble Space Telescope. This was an incredibly interesting mishmash of engineers and software and computer programmers and geneticists and molecular biologists. That's what it took to build a company like this, and they were acquired by Roche, the big Swiss pharma company and diagnostics company, shortly afterwards. And then, for good measure, they decided to turn their attention to human genome sequencing. And not far from here, 3 hours from here, down in Houston at Baylor College of Medicine, in May 2007, I was watching with some incredulity as Jonathan Rothberg here, shown on the right, presents the one and only James Watson, the most famous scientist one can think of, with his personal genome, his personal digital genome on a portable hard drive, for the first time that any human being had received their complete genetic code using one of these next generation methods. Venter was still getting his genome more or less tidied up using the old Sanger method, but this was the first fully using next gen sequencing, and that was memorable. And as I watched that, I thought, right, I'm writing that book. This is the trigger. This is what has convinced me there's a book to be told here. And here is Jim sort of backstage at Baylor College of Medicine with Richard Gibbs, the director of the Genome Center, James Lubsky, a world-famous molecular geneticist, and other members of the team are looking over the highlights of his sequence. Watson had agreed to be sequenced with two conditions. Number one, he would release his entire genome to the public. He would have Cold Spring Harbor, where he lives and formerly worked, build a website where his entire genome could be made publicly available for students to look at. But there would be one caveat. There's one gene he didn't want you to know about, nor did he want himself to know about. There's a gene on chromosome 19 called APOE that in one particular form or flavor can dramatically increase one's risk for Alzheimer's disease. And although Watson is in great shape physically and mentally, he had a little bit of family history and just did not want to be concerned that he might, and start worrying that he might have a risk variant in this particular gene. So they wiped out that gene from the record, and that might be where the story ends, except that it turns out, just in the way that back in London you could find markers that segregate with genes, well, the neighboring genes are more often than not going to segregate with that APOE gene. So whatever version he has, if I study what he's got around it, I can probably make a decently educated guess as to whether he's got APO2 or 3 or 4 and what the implications for his health might be. So when this was pointed out publicly at a conference, not unlike this one, some years back, Cold Spring Harbor guys very quickly ran out and ordered a whole bunch more genes be redacted from the record, so that now it's impossible to really look into that part of the record. Jim has been celebrating the 10th anniversary of the Human Genome Project. I'm sure there are a whole bunch of stuffy black tie dinners in Cambridge, England. There are some more fun, frivolous parties in New York, and the person who posted this photograph on Twitter described his 4 companions here as his 4 nucleotides, which, I don't know, I think that's quite nice, but I wish I'd come up with that line, but you see him with Brian Cox and Alan Alda and a number of others. So for a while it looked as if 454 was the company to beat, but around about 2008 the tide began to turn. I think 454 pretty much reached the end of their sort of runway. They were able to sequence very small genomes, microbial genomes, but not really, other than Watson, which cost $1 million, still better than $1 billion, but still not exactly cheap. Not a method that was going to be used for whole-scale, whole genome sequencing, it would appear. And then in November of 2008, these 3 papers appeared, all in the same issue of Nature, each of them describing a human genome. There was a cancer genome, there was an Asian genome, and there was an African genome. And we in the community had never seen anything like this. This was staggering. It used to be when I was publishing genetics journals, if you got 3 papers describing 3 single genes linked to a disease, you were ecstatic, you were writing press releases and begging the New York Times to cover you. Now these are 3 genomes back to back to back. What they all had in common is that they were all using that sequencing system developed by the Brits in the pub, the Solexa sequencing method, except now the blue boxes that formerly said Solexa now said Illumina on them. And it was John West, who is related in ways that you will hear later, to our next speaker, Ann West, who was the CEO of Solexa, who engineered the sale of Solexa to Illumina for $650 million in 2007. It sounded like an awful lot of money. That was the steal of the century. Here is Shankar Balasubramanian in a very recent photo celebrating a paper that got a lot of press in the UK. Just as a side note here, he's discovered a new interesting form of DNA. I show this because this is a mural in his office. So clearly, the people at Cambridge University have looked kindly upon Shankar and all of his success and built him a bigger office where we can actually have a group meeting now. So that's nice. Don't worry about the units of this. This graph simply shows 2 things. One, the price. This is 2005, 6, 7, 8. As the price of DNA starts to tumble down, the number of people being sequenced starts to grow inexorably, if not exponentially. First woman sequenced. This is not data that's been published, but she certainly got the recognition for being the first female to be sequenced, was a Dutch clinical geneticist named Marjolein Kriek. And she was sequenced because her name sounds like Crick. And the Dutch thought, well, if the Americans are sequencing Watson, we should do Crick, right? And they just so happened that they had one in their midst, and so they sequenced Marjolein. She's was happily just working in a medical center, wasn't seeking fame and glory, and suddenly there she was. They were building statues for her and photograph exhibits and so on, and she was even immortalized in sort of the Dutch equivalent of Doonesbury, and even the Dutch couldn't quite figure out why she'd been, why not Scarlett Johansson or somebody really important. But a couple of years ago, I'm pleased to say, I was finally able to reunite Crick and Watson, so that was good. Here is our next speaker. I'm not going to say much about this, but I want to point out one important thing here, which is this was at a symposium held in Boston a few years ago, I think, where I first met Anne. And she was talking about the first series of analyses that she was doing about her own genome and that of her family's genome. And not to steal her thunder, but that is the doctor's note that led to Anne having her genome sequenced. And I thought that was impressive, and I'm so glad, and I've lived off this for many years now, that she was gracious enough to share that with me. I show this because this was the company Illumina that was starting to offer now personal genome sequencing. But rather than risk sticking a thumb in or poking a thumb in the eye of the FDA and not requiring a doctor's note, they were very cautious and said, you know what, we're going to require a doctor's note. This was at a time when consumer genetics was starting to come to the fore, and that's fairly significant. I'm just going to spend a couple of minutes on that before I get back into the sequencing and the medical stuff. So in 2007, the other major event that made me want to write the $1,000 genome was the launch of this company. This is 23andMe. The co-founders, Linda Avey, on the right in the top in this figure here, and Anne Wojcicki, who is married to Sergey Brin, the co-founder of Google. And so this raised a lot of sort of concerns, like Google's getting into the genomics game. What the hell are they going to do with my data? Here's Peter Gabriel. Here's Peter Gabriel being one of the first guinea pigs for this. Now, this is not a sequencing company. What these guys were doing were taking saliva samples, extracting the DNA, and then running it over a device that Illumina had built a very successful business in before it sort of morphed into the sequencing game, called a DNA chip. And what this does, it doesn't sequence all the billions of bases in your DNA. It just it interrogates about 1 million of them. And obviously, these are chosen for parts of your genome that vary from person to person. and typically lie in or close to genes that may be of medical relevance or maybe relevance for other things as well. This is what some people think of genetic testing. This is from the great XKCD cartoon strip. Are my tests back? Yes. Yes, we can't be sure about this, but we've analyzed genes on several of your chromosomes, and it's hard to avoid the conclusion that at some point your parents had sex. There was a lot of frowning, particularly in the medical establishment, that this consumer genetics was just a load of rubbish and you'd be better off taking your $200 and spending on a gym membership. What the heck are you going to learn by spitting in a cup and getting some website telling you you've got a gene for this or a gene for that? There's a huge amount of skepticism. Because you're going to hear a lot about what you can learn from a personal genome, I'm only going to very quickly show 3 photos for what I learned when I did my 23andMe results. They tell you a lot of information about your ancestry. They can divide your DNA into regions that are inherited that most closely resemble African origin, Asian origin, or European origin. My family is exclusively from Wales, so predictably I get 100% European, which is terribly, it's kind of boring actually, but that's what the data says. I'm the proud owner of 2.3% Neanderthal DNA, and so are you, let me assure you. In fact, you may have more than that. We are all walking around with the small traces of Neanderthal DNA, and 23andMe will even print a custom T-shirt that says to the tenth of a decimal place what percentage of Neanderthal DNA you carry. But most people will go to this for some information about their genetic risks. One of my interesting risks that came for more complex diseases came back that I carry twice the lifetime risk for age-related macular degeneration, which is a disease that my mum back in the UK was diagnosed with last year. And now I'm going to my optician or optometrist and saying, look, tell me, how do things, I also learned that I carry a gene. It's asymptomatic because this is a recessive disorder, so you need 2 copies to actually have the disorder. But I'm a carrier, as are many people I've met on stages like this, giving presentations for a disease called hemochromatosis, which is when you have high levels of iron in the blood. And this was sort of, not amusing, but kind of interesting or ironic to me, because I published the paper describing this gene in 1996 when I was an editor with Nature Genetics. So that's a strange, interesting claim to fame, I suppose. 23andMe has done a lot of interesting things, taking all of those data. They have about 500,000 people have done this test now, and using it to map and potentially identify novel disease genes. If you've got DNA material from 5,000 Parkinson's patients, you can now look at all of the variants and all of the patterns in those individuals compared to controls and see which markers are kind of clustering with a risk for Parkinson's. And using that, and this is all in a top-tier peer-reviewed journal, they've been able to make important breakthroughs, I think, in Parkinson's disease and asthma and some of these very difficult diseases where it's hard to separate the genetic component from the environmental issues. But as you may know, last year the FDA finally clamped down on 23andMe. 23andMe were running TV commercials and really trying to push to get 1 million customers last year. And that's when FDA gets nervous. It's when for all this time, no doctors have been required in the delivery or the analysis of these genetic data. But when you start running TV commercials and people who perhaps aren't as sophisticated as some people who are going to ferret out a 23andMe start seeing this and being swayed by the messages of health and well-being that you're seeing on your TV screen, then that's when FDA starts to get a little nervous. And they worried in this letter about scenarios such as women receiving results for a breast cancer gene, for example, that may be inaccurate. Maybe it's a false positive, but they might do something very rash and very draconian surgically, obviously with the guide of a medical professional who perhaps doesn't understand what these tests are all about. That was one scenario, not a scintilla of evidence that this has ever happened, but that was what FDA was concerned with. Or the opposite, that you may get a negative test for one of these genes and start thinking, hey, this is good, this is great. I don't need to worry about screening for breast cancer or prostate cancer anymore, without completely understanding that these tests aren't completely 100% conclusive. We can't pretend that we've isolated and analyzed every single gene that has a variant that may impact your risk for cancer or heart disease or hypertension or so on. So at the moment, 23andMe has been slapped on its wrist big time, and while it's still in business and still offering its test, and you can still download all of your raw data. The FDA is not interfering with that, but they are blocking any of the interpretation that goes along with it. You can still get your ancestry results and so on. And the other thing I would point out is that decision by FDA, which I think was an overreach, goes in the face of empirical data published in the New England Journal and elsewhere that says that people are very able to handle and interpret this information. And for the record, I think 23andMe did a brilliant job in educating the public about what these tests can tell you and what their limitations are. So, but for all of this progress in genome analysis and genome sequencing, how is this going to impact medicine? Let's get back to the theme that we heard about this morning. And so I want to introduce you to a young boy named Nicholas Volker, who has become the poster child for genomic medicine, certainly when it comes to the benefits of genomic sequencing. You heard this morning about fenestration, about these sort of tiny holes. I never would in a million years thought you'd hear that term at this symposium, but that is what Nicholas had. He had these tiny holes in his intestines that, as fast as the doctors tried to sew him up and repair them, others would emerge. And for many years, in his early life, 2, 3, 4 years of age, he was very, very sick, almost living in hospital, scores of surgical procedures where they simply ended up where they started. And in the summer of 2009, one of the medical doctors caring for Nicholas wrote to more of a DNA expert in Wisconsin, where he was being cared for, and said, his name was Howard Jacob, the recipient of this email, and the letter went, Howard, any chance we can sequence this kid's DNA? Because frankly, there are no drugs we can give him, there's no more surgeries we can do, we're running out of time and we don't know what else to do. This is Howard Jacob, and he said, you know what, yeah, let's do it. This is still early days in genome sequencing, but he had a partnership with 454, the company that I mentioned a little earlier. And rather than try and do that whole genome, because that would still have been on the order of about $1 million, I showed you remember the figures for Jim Watson's genome, they took a shortcut, and that is they looked at all of the genes in your genome, but ignored all of what some people call junk DNA, all of the bits, all of the sort of the deserts laying in your genetic code that have just grown up over evolution and have little, well, I wouldn't say little or no function, they have unknown function, but for the most part, big asterisk there, they're not known to be of medical relevance. So you can look at just the genes, and we call that the exome rather than the whole genome. So they looked at the exome of Nicholas, cost about $75,000, and they found, if you will, a sort of a ticking time bomb. It wasn't immediately, oh my God, there it is, there's the mutation. They had to look at other patients that have had this mutation, they had to talk to doctors, they had to do other tests. But the more they looked at it, the more they felt there was a connection between a mutation in this gene and that could explain some of the sort of the autoimmune disorder that Nicholas was suffering from. More critically, in terms of his immediate medical care, there was a technical term for the disorder represented by this mutation, which I can't pronounce, hemophagocytic lymphohistiocytosis. He needed a bone marrow transplant because patients with this very, very, very rare mutation cannot clear Epstein-Barr virus, and if you're living in a hospital most of the time, one imagines you're going to be exposed to that sort of thing. So finally, the doctors had a reason to put in the medical report, we have a diagnosis, and we have a treatment. And so we did the treatment, and fast forward to a picture 2 years ago, Nicholas is now home, he's back at school, he's a healthy, rambunctious child who is the apple of the eye of all of the doctors at the Medical College of Wisconsin and Children's Hospital who've cared for him. Not completely out of the woods, still complications, still hospitalized from time to time. But this is about as good a story as one could imagine that shows that it wasn't just a matter of getting the price of genome sequencing down to an area that is compatible and similar to other expensive medical tests, but it's now that we have the computational facility that we can put all of that information to a computer, we can run programs, we can run filters, and we can screen and look for all of the thousands of areas that his genome might differ from what we call the reference genome. We can zero in on those that are known to occur an effect to mess up a protein and are known to be what we call deleterious. And then we can look at the proteins themselves and figure out, well, which ones seem to make sense in the context of this disease, and so on. And you can apply all these filters until you can with increasing certainty, find the mutation that you think is the causative one. And I could spend the next half an hour going through examples of other patients who have had these sorts of stories where, at the very least, they've been able to finally get a diagnosis. They've been able to end their diagnostic odyssey, and they've got peace of mind, and they can, in some cases, there's a therapy, in some cases, there's not. Sometimes there's just the hope that maybe my child can go into a clinical trial, maybe there's a gene therapy around the corner. In this case, this child, although in retrospect, I think that the gene was perhaps could have been found by other means, but she was sequenced at a major research center in Phoenix, Arizona, called TGen. And Shelby Valint, who is a prolific YouTube video maker, can now walk. And there's a video online at this YouTube URL here, bit.ly/ShelbyV, which will show you what she looked like before. It's a dramatic turnaround thanks to the efforts of genome analysis. While I give all kinds of credit to the group at Wisconsin for really blazing a trail in this space, you should, I was worried, I told Ben before, if I mentioned Baylor, am I going to get booed off the stage? Here, I don't know quite what the rivalry is within different Texas institutions, but this was a paper in the New England Journal of Medicine from a group at the Baylor College of Medicine who have sequenced more of these children now, looking at their whole genome to end these diagnostic odysseys. These are children with diseases that are severe, potentially life-threatening, and for which the best medical minds cannot find a diagnosis. If we could look at a child and say, oh, they have spastic paraplegia type 3B, and we knew the gene, and we could just isolate that gene and sequence that gene, we'd do it. But that's the whole point. We're saying, well, we think this child has this disease, so we'll look at that gene or the family of genes, and then that turns out negative. Meanwhile, you're incurring thousands in hospital bills, and now you say, okay, well, what other genes might be responsible? There's got to be something. And the point I'm trying to get across is that now a whole genome approach can get all of that information. It's just we cast the net wide enough, we know that somewhere in there, somewhere in our computer, is the answer. And now we've just got to have sufficiently skillful bioinformaticians and geneticists and so on to extract that answer. So this becomes more of a computational problem now than a medical problem. We're looking for evidence that one glitch or one deletion in that patient's DNA is causing this particular disease. This is Eric Lander. You saw a photo of him this morning. He's the director of the Broad Institute and was one of the chief architects of the Human Genome Project. He has probably overseen the sequencing of more human beings than anybody in the world. And yet when it comes to his own genome, he is surprisingly squeamish and says, oh, I don't know if I want to have my genome sequenced. But in a quote that I've taken from my book that he mentioned a few years ago, if God forbid I had cancer, I would get my cancer genome sequenced in a heartbeat. And to do that, he co-founded a company. I'm not endorsing any companies here, but these are the companies that I think are potentially showing paths the way that medicine is going. This is a company in Boston called Foundation Medicine. They went public last year. That looks at if a doctor from anywhere in the country, or perhaps anywhere in the world now, has a cancer patient, and as you heard this morning, I love Maro's phrase, cancer is like malignant snowflakes. Everyone is different. Everyone builds up a different pattern of mutations, and you've got to figure out which are the driver mutations and which are the passenger mutations. How do you find the driver mutations? Well, one way is you take a biopsy of the tumor, you send it to an outfit like Foundation or to the cancer clinic at your big hospital. I'm sure Baylor is doing this, and other major medical centers in Dallas and elsewhere are doing this as well now, or if they're not, they're going to start very soon. And you sequence, maybe not the whole genome in that patient or the whole cancer genome, but you look for you sequence the 300 genes that we know from all of the papers that have been published in cancer over the last 20 years that are linked to different types of cancer. And then the result that a company, an outfit like Foundation will give you we'll send back to the oncologists and the medical teams, here are the mutations we found, here are the drugs that we think best fit the molecular profile of your specific patient and his or her personal cancer. And here are clinical trials with more experimental drugs that your patient may be available for. This service was offered for solid cancers, breast, prostate, and so on, liver, pancreatic, and now they're applying it now for leukemias and lymphomas, so-called hematological cancers. So cancer is, in order to understand cancer and be able to prescribe the right drug or drug regimen for your patient, you've got to know what is going on at the molecular level. And we're now seeing the ability to do that. So I love this quote. We're close to the $1,000 genome. It's now the analysis, as I've been hinting at, that's sort of the problem. It's not $1 million. It's not anything close to that. But it's the actual sequencing of your DNA or my DNA, if I should get sick, that's becoming relatively, we're not out of the woods yet. We can still improve the accuracy. We can still improve the speed and the price. But it's now about how we interpret it. And putting this into a medical system where many doctors are really scared of DNA. Whatever they learned about genetics in medical school was ages ago. This field, as you've seen, it's changed so fast. Genomics has really only come of age in the past decade or so. And you've got, of course, all of the insurance and regulation and the health IT and all of this stuff, and that's the subject for another Brown Symposium maybe in a couple of years' time. The good news is, just thinking about ending diagnostic odysseys, however, is that so far, based on published records from Baylor and from the Wisconsin team, is that a number of health insurance companies are taking a very enlightened approach to reimbursing this whole genome sequencing because they see the benefit not just medically but economically. They don't want to be covering patients for weeks or months at a time in hospital with medical procedures there's an element of guesswork to them, and genetic tests that are often patented and cost thousands, if not tens of thousands of dollars to run, when you can now just put that DNA in a machine I'm going to show you in a second and get the whole information for just thousands of dollars. This is what the cost of a genome sequence was last year, about $5,000. That was the price I was told by the chief science officer, David Bentley, at Illumina. He should know, because this is all happening on his machines. And Illumina, about last year, maybe a couple of years ago, started running a conference called Understand Your Genome, so that if anybody here was really, really, really interested in getting their personal genome sequence, you can now, $5,000 is a lot to pay to go to a conference, but for $5,000 you hear 2 days of fantastic lectures, and then you're presented with an iPad with your personal genome sequence on it, because you've presented a saliva sample in the weeks running up to the show. So that is what's being offered. Maybe it's a bit of a loss leader right now, but maybe not. This is the falling cost of genome sequencing compared to Moore's law. That's you heard about that this morning. That's the classic trajectory highlighted by Gordon Moore that explains the doubling capacity or the halving price of your laptop year upon year upon year, or your iPhone or whatever. Until for the last sort of year or two, we were at this sort of sub $10,000, $5,000 plateau, and then suddenly, boom, it dropped to the $1,000. So why was that? Well, that was because 3 weeks ago at the JP Morgan Healthcare Conference, Illumina's CEO, Jay Flatley, introduced this box. This is called the HiSeq X machine. And this machine, this is the new top-of-the-line sequencing instrument in the fleet offered by Illumina, can now sequence a human genome for $1,000. This requires a little bit of creative math, and President Berger will, I'm sure, be scribbling and commenting on this later. But in order to get that calculation, you've got to buy 10 of these machines. Illumina will only sell them in groups of 10. That's $10 million. This is not cheap. I didn't say it was. You've got to have some deep pockets to get this going. But if you install those and you run them non-stop, because these are very thirsty machines, in terms of processing DNA, and you run them non-stop for 4 years so that you can depreciate the cost over those 4 years, then the cost of doing a genome comes out to about $800 for the chemicals, about $137 per run for depreciation, and the little bit of sample prep that you do up front before you put the DNA on the machine is about $65, and lo and behold, you come to the magical $999. Yes, they've left out a couple of things. They conveniently left out all of this. How are you going to store all of this digital data that comes off? Well, if you can afford this, you've got a state-of-the-art data center as it is, but let's just put that to one side. They also conveniently left off the cost of electricity. It'd be nice to run these machines, with the lights on, and they didn't. If they put that in, that might have just put it over the $1,000. But that's where we are, and that's very exciting. And so when I saw Jay last week at Stanford, I had to grab the CEO of the $1,000 genome, or at least the proclamation of the $1,000 genome. But I've reason to believe, certainly the markets and the community has taken that announcement as seriously as anybody else leading up to this moment. So where does that leave the competition? Well, here's Rothberg. He's now laying low. He left. He started another company to do sequencing, an extraordinarily interesting company called Iron Torrent, which is now part of Life Technologies. Iron Torrent was a company in which Jonathan Rothberg realized something that had not been widely appreciated, which is that you can measure DNA sequence by pH. This sounds absurd, but every time a building block is stitched into a molecule of DNA, a proton, a hydrogen ion, is released, and that's the unit of acidity. And using special sensors, you can detect that transient increase in acidity, and thus detect when a building block has been added, and thus calculate the sequence. And that was touted by Forbes a couple of years ago as the next $100 billion technology business, but now they're facing very, very stiff competition from Illumina. This is, I think, going to be the next big thing, or little thing, in DNA sequencing. And I'm actually carrying around with me people can come up later or in the break and have a look at this. This is a DNA sequencer I'm walking around with in my jacket pocket. This is the MinION, that was unveiled 2 years ago by a British company called Oxford Nanopore. So finally, Oxford is getting back in the game, I'm delighted to say, after all of that Cambridge nonsense. And this is Clive Brown. This is the guy who, when he was at Solexa, sent around that email saying, "We've done it." He's now turned up at Oxford Nanopore with one of his other key colleagues, John Milton, and they are trying to capture lightning in a bottle again. This is not going to do the $1,000 human genome. This is not even commercially available yet, but it looks like it is on course for release later this year. And the fact that to run a DNA sequence, all you need is this little cartridge, a USB cord, and a laptop. That is what I call disruptive technology. I mean, this will be the highlight of every lab from Southwestern to wherever you care to go. How does this work? Very briefly and quickly, this is based on something called nanopore sequencing. This is the concept back into the nano field now. This is a naturally occurring bacterial protein with a channel down the middle that conveniently fits a single strand of DNA. And so if you can coax a single strand of DNA using another enzyme to fall through this pore, and you apply an electric current across this membrane, this surface, and you measure the current, or more accurately, you measure the resistance using some very clever informatics that I don't begin to understand, you can translate the resistance, or the current, to the DNA sequence, because each of the 4 building blocks of DNA, A, C, T, and G, have different sizes and shapes. And if you do this often enough and teach the programs to learn using hidden Markov models and things of this sort, you can begin to produce the patterns and the information required to render these squiggles, as Clive Brown calls them, into actual DNA sequence. And the first glimpses of this technology, I think, are going to be presented at a scientific conference in Florida next week. So this is a very exciting time for our field. So whether it's Illumina or Oxford Nanopore or IBM or whoever it might be, some company I've never heard of, it could be a company in China or Japan, busy about to unveil some breakthrough technology, in a way, it doesn't really matter. The point is we are reaching the point that genome sequencing is becoming cheap, incredibly accurate, incredibly powerful, and even reasonably conservative but hugely respected medical geneticists like this guy, David Valley at Johns Hopkins, are saying that whole genome sequencing will become the standard of care. We'll sequence somebody once and we'll read it as often as we have to in order to make medical decisions during the course of their life. We could do this at birth. After all, every newborn, it varies from state to state, but we prick their heel, don't we, with this thing called the Guthrie heel stick test. We take a little drop of blood and we analyze it for 20, 30, 40, maybe 50 different genetic disorders such that we can make a quick dietary intervention rather than risk some deadly inborn error of metabolism affecting the child in those first precious few days and weeks of life. So we're already doing newborn screening, but why should we stop at 20 or 30 or 40? We can discuss. And while I focused on genome sequencing, I want to quickly point out two other things. One is this technology is being applied for many other things. So how about the prediction of Down syndrome? Down syndrome, caused by an extra copy of chromosome 21, is typically something that we have to do a very dangerous, well, not very dangerous, but a somewhat dangerous procedure of amniocentesis or something of that sort with risk to the fetus's life. In a small minority of cases. And now we can detect this chromosomal material just by taking a blood sample from the mother. And there are now some 4 companies offering this test. And indeed, this photograph taken from a friend of mine, this is how birth announcements are done on Twitter these days. It's not just, hey, we're having a baby, but look, it's chromosomally normal. We're good for 21, 18, and 13. The other thing I wanted to mention in that context, which I think is a fascinating but a completely different talk, is the microbiome. There's a huge growing interest in all of the bacteria that are in our gut and on our skin that are increasingly being shown to impact our health. And I don't have time to get into that, but that's another fascinating subject. This is my final slide. This is the FARGen project. There's a tiny island or set of islands called the Faroe Islands, nestled between the northern coast of Scotland and Iceland, with some 50,000 inhabitants. They're sequencing the whole country here to put that information into their medical system and change healthcare in that country. And one would like to think that if they can do that in places remote and sort of, I shouldn't say behind the times, because I don't think they're anything like behind the times. If they can do that there, you would think that we can start to think about these sorts of measures in this country, and maybe we will. So it's been a pleasure meeting students here and talking to you. I hope you enjoy the rest of the conference, and I'm happy to take questions. Thank you so much.
So we have time for some questions. If you have a question, raise your hand. We'll try to get a microphone to you so that everybody can hear your question.
This one.
Hello.
Hello.
I was wondering, as the cost of sequencing genomes decreases exponentially, I'm back here.
Thank you.
As the cost of sequencing genomes decreases exponentially, and as our processing power used to understand things like genome-wide association studies increases exponentially, do you think our ability to understand the human genome can increase exponentially?
That's a very nice question. I suppose the short answer would be no, because I think there's still an awful lot, and I obviously glossed over this, there's an awful lot about genetics and the genome and its link to human biology that we don't understand. And so you mentioned these things called genome-wide association studies, which have been all the rage in the community for many years now, where people are taking large subsets, large cohorts of patients with different diseases and looking at tons of markers and trying to find genes that are highly correlated with Parkinson's disease or Crohn's disease or every type of disease that you can imagine. But so far, despite the very best groups in the world with the very best technology, we only seem to be capable of explaining a very small amount of the heritability of these different diseases. Obviously, these are very complex. People are finding dozens of genes associated with asthma and Crohn's disease, and not to mention diseases like schizophrenia and manic depression and so on. And there's a bit of a missing link here between what we know so far today, 2014, at the genetic level, and how we can translate that into predicting exactly who's going to get sick, because we haven't spent enough time or nearly enough money on understanding the environmental component, or the allied ancillary components like I just mentioned, like the microbiome, and the impact of microbes in the environment and in us on our health. So we've got a long way to go. That's why basic research is so important. That's why increased funding for NIH and philanthropic research is so important. So I'm optimistic, but I don't know that the science necessarily can grow as fast as the technology can.
Thank you.
Hi.
Obviously, one of the big subjects of your presentation has been how genomic sequencing has become so much easier, so much more efficient, so much more cost effective,
Yeah.
and how now we're approaching the $1,000 barrier, which is the subject of the book that's
Yes.
How much time do you think it would take before we can even push that barrier further and say that genome sequencing becomes so easy and so cost effective that it becomes sort of standard that everybody can have it at little or no cost potentially?
Yeah. You really want me to put a year on it? 20, I don't know. Maybe, I could say 2024 or something like that. I would say a couple of things. Firstly, I didn't see you'd think I would know, but no one told me. So I didn't see this $1,000 genome announcement calling. I knew that the Illumina CEO was speaking at this JP Morgan. I went to my computer. I wasn't at the conference. I tuned into the webcast. And I'm late, and suddenly I hear the magic words, the $1,000 genome. I think, oh my God, I turn on Twitter and I'm tweeting his remarks as fast as I possibly can to get the word out. It was exciting. Even if it makes I won't finish that thought. So I think that's good. But as I showed in the picture of that technology, this is involving dyes and cameras. There's still a lot of very expensive components to what is currently the state of the art in DNA sequencing. And the beauty of this nanopore device is that while I'm not minimizing or trivializing the huge amount of work and labor that has gone into engineering the enzyme and the substrate, and I can't even tell you that it works right now because I'm not privy to any private data. But assuming that works, now I've got a sequencer that not only I can carry around in my pocket, but it just requires DNA prepared, according to a certain recipe, and I would drop it in, plug it into a laptop. Obviously, I've got special software loaded on the laptop, and I'm away. And whether it's Oxford Nanopore or there are many other nanopore-type companies working along similar lines, I'm sure in a couple of years there's going to be an almighty IP patent battle as they figure out, who can sue who and get rid of the competition using that route, which is all too common in this field. And then, while the portable thing is lovely, but what about now growing that and building a sort of a machine like the HiSeq X10 that I showed you that can harness, a million of these minions or whatever it might be. So I can't really safely predict a date for when that will happen, but it's not going to stop at $1,000 because, some of the most brilliant, most innovative scientists are really hell-bent on this field, and whether it's nanopores or something, that's the best I know of today. There's going to be something that I haven't thought of, or anybody here has thought of, that probably in 10 years is going to bring us the next step to what you're saying, which is essentially cheap, ubiquitous genomes. Thanks for the question. It's one. Okay, go ahead.
This is a little bit maybe out of your thing, but it seems like to me, getting the defective gene in has really lagged. I mean,
yes.
Does that make sense?
Putting the healthy gene back in.
Yes, sir.
Yes, absolutely. Gene therapy.
Seems like it's kind of hit I think you had a disaster at the NIH and so forth.
You did.
Yeah.
You did. It was actually a Penn, but it was funded by NIH. So yes, there was a death in a highly publicized gene therapy trial. This is where you know what is wrong with a patient, and you isolate that gene, the healthy version of that gene. You put it in a virus or some sort of vector. You hope the virus is completely innocuous and benign, and inject it, and you hope that the healthy gene is taken up by the cells that are carrying the mutation and gets reinserted, or maybe not, but that will then provide the all-important enzyme function or whatever is missing and has been compromised in the patient's genetic code. The problem is you're putting a virus that is not necessarily benign. And if you put in a high dose, and this is what happened with the case of a young teenager volunteer named Jesse Gelsinger, an immune reaction, a devastating immune reaction takes place, and the doctors weren't able to stop it, and he died. And I would like to think that his death has not been in vain, and that it has helped introduce new standards in the gene therapy field. And the good news is that now reports are increasing of patients with other diseases, particularly sort of leukemias and things of this sort, and other autoimmune diseases, being treated successfully. One always hesitates to use the word cure, but being treated successfully using modifications of this gene therapy protocol. We've learned an awful lot in the last 10 or 15 years. And I think, just to kind of speculate a bit further, the hope for this field, or one of the hopes for this field, is that we won't have to take a virus and inject a gene into a patient. We can actually use some molecular toolkit to engineer the repair in the patient's DNA. And there are a number of biotech companies, I won't mention them now, but we can discuss offline if you want, that are sort of pioneering this this sort of approach. And the toolkit for genomic engineering is undergoing an explosion right now in the basic labs that is inevitably going to spin over into the biotech and pharma community. So it's going to be, the next 10 years are going to be really exciting in that field.
Thank you. There's time for about one more question.
Right, okay. Hi.
Hi. My question is regarding the constraints on transferring the knowledge and deciphering what genes actually have repercussions on our health. Do you believe that a large part of it is just like absence of a large enough database with like, I guess like the genes of the population and like their health indications? And if more people were to like be tested, be analyzed, would that actually help bridge that and just like be able to get and make those connections between like the genes and the diseases or the conditions that might be correlated to?
But it allows us to sequence many more people. These could be volunteers in clinical trials, or they could be populations gathered by countries. So I mentioned the Faroe Islands sequencing their population. The Saudis are going to sequence 100,000, I don't know if they're patients or just members of the population. A biopharmaceutical company called Regeneron just announced that they are going to sequence 100,000 patients in concert with a health insurance company, presumably because they want to apply this technology to run better clinical trials. And these HiSeq X10s that I just mentioned, the last one that was sold, they're only planning to sell 5 this year, and the 5th one just got snagged by a subsidiary of Amgen, one of the very first and most famous biotech companies in California. And I know the guy who runs the research there, a guy named Sasha Kam, a friend of mine. He's the guy who discovered the BRCA1 breast cancer gene when he was with Myriad 20 years ago. He told me a few years ago, I'd love to do this genome sequencing thing, but the technology's not quite there yet. And then just the other week, I saw that press release that they're ordering that. So that tells me Amgen has decided, that's it, we're ready. We can now analyze the genomes of all of our patients because they want to improve the success rate and the safety of their drugs. The last thing they want is to put a drug on the market only for sort of an adverse event or a series of side effects to suddenly come to light. If they can screen those out before that happens, then they benefit and the field, we benefit. So I think more genomes, more.
Thanks.
So, thank you all very much indeed.